So my name's Phil Thomas. I'm a PhD student at University of New England in Armidale, and I'm working on this project which is looking at the microorganisms that live around the roots of hydroponic tomato plants. So we're switching from the broad scale to the microscopic, so hold on to your hats, do not adjust your sets. So this is a project which is part of the future farming program of the Future Food System CRC. It's a collaboration between Costa Tomato Exchange at Guyra and UNE and Western Sydney University. And our ultimate aim in this project is to develop a test for the microbiome in the hydroponic systems that the growers can use to track the health of the microbiome and hopefully get an early warning of any problems appearing in the crop. So we started the project back in 2020. One of the first things we did was to set up an experimental crop in the UNE glasshouse. And we aimed to make an environment which was as much like a Costa commercial crop as we could. So using commercial varieties of large truss plants and grown to maturity and fruiting before we started our study, and growing in the same kind of substrate, using the same kind of irrigation and maintaining the crop, picking and leafing and etc. Because all of these factors, the environment, the plant, the maturity of the plant, have a big bearing on the makeup of the microbiome. So we also study crops in the Costa glasshouses. So in 2021, we sampled a commercial crop again of the large truss tomatoes, which is the most economically important for tomato growers. And we sampled it for the whole crop lifetime, starting in January with the baby plants and finishing after the ethylene addition and just before the final harvest in November. So 16 time points, 500 something samples. And this crop turned out to be a particularly successful, healthy crop. So the data that we've extracted from that now is our reference for what the microbiome of a healthy large truss tomato looks like. So we also, this activity is just finishing up in the UNE glasshouse right now. So in contrast to the healthy crop, we're looking this time at the microbiome response to tomato plants that have a pathogen problem. So again, this is the same kind of environment, large truss plants and grown to maturity before we started treating them. Started last year, and around the end of last year, I started trying to infect powdery mildew into the crop. It turned out to be a little bit different, a little bit difficult. And just as I was giving up after the second inoculation, we managed to get powdery mildew into that crop. And we also then took some Fusarium pathogen spores from one of the Costa glasshouses and managed to get a infection symptoms coming up in the crop as well. So the samples of this experiment over this time are still in being processed. They haven't been, we haven't done the DNA sequencing yet, but this will be an important contrast to our healthy crop. So what have we found about the hydroponic microbiome? Well, one thing is that it's highly consistent throughout the glasshouse, which is, as you would expect, in a highly controlled, protected environment, and it changes gradually during the lifetime of the crop. That's also what we expect, because the plant development and changes in the environment affect the microbiome. But the fact that it's a consistent and gradual change is good for our test, because the more consistent the healthy signal, the easier it is for us to detect deviations from that. The other thing we found that was expected is that there are distinct communities living in different locations in the slab, and that's as a result of different environmental conditions, pH gradients, EC gradients. And the fact that we can clearly distinguish those is also good for us, because it shows that we can detect changes in the microbiome coming from the environment. Now, when comparing to soil, we found something that we didn't expect, and that is that there were more species in the hydroponic system in the root zone than we found in tomatoes growing in soil in a high tunnel system. So similar kind of commercial large truss tomatoes. But there's a different balance between the two systems. And there's also a very big difference in the type of organisms which are dominant in the two systems, which you would expect. So the hydroponic system is not what you may think a clean, not very many organisms, not much variety. It's actually as complex as a soil rhizosphere, but with a completely different environment, a completely different makeup, and very different organisms. So possibly worth investigating. Now, the approach for our health test is based on the principle that the microbiome is closely attuned to the plant, especially in the hydroponic system, where all of the nutrients for the microorganisms are coming from the same kind of plant, the same crop. And if the plant is affected by stress, nutrient problems, pest, pathogen attack, it can change its root chemistry, and that affects the makeup of the microbiome. So if we can detect reliably the signals that indicate the deviation from a healthy microbiome, we can give the early warning to growers, gives them the opportunity to do some increased surveillance, early intervention, and a week or two early warning of problems in the crop is a very big advantage in the hydroponic system, where the difference between snuffing out a problem before it spreads, before it gets out of control in the glasshouse, makes a huge difference to the overall yield of the crop. So that is, in a nutshell, the hydroponic root microbiome project. So thanks everyone for joining the presentation. Thanks everyone who's been involved in this project, and happy to take any questions.